Hi, so my name is Bassi. I work in Weizmann in the lab of Karina Yaniv for 6 years as a postdoc. And during this time, I conducted two projects. One was related to blood-brain barrier, and the second one, in collaboration with Idea Biomedical, we studied a lymphatic disease called Kaposiform Lymphangiomatosis. That is a severe disease that affects children, and there are no currently treatment for this disease. So using as a model Zebrafish, our small fish, we managed to mimic the human feature and physiology of this disease, and we studied we were able to identify some new drugs using the Hermes system that are efficient in treating this disease and can reverse the phenotype in the fish. I mean, I used to work a lot with confocal microscopes. The advantage of Hermes is it's really fast and the quality of the pictures is good. So for our purpose, that was to have like a sort of high throughput screening, because we need to test a lot of drugs to identify the good ones. And in a relatively small amount of time, it was perfectly. It was fitting perfectly the aim of our project. We needed to image the fish very fast because the fish can die. We couldn't run the project for so long. So the fact that it can acquire in 20 minutes an entire plate, it was amazing for us. The system is very fast in acquiring the picture. So I think that 96 well plated took something like 15 minutes, 20 minutes to get the entire to take pictures of the entire fish in the plate. We got like 2-3 plates with drugs for each experiment, so it took us usually 1 week to analyze and generate the data that and the graph that we need for the project. Let's say we run 2 or 3 plates per weeks, and this allow us in few months to test the big amount of drugs that in the end we tested during the project. So before using the Athena software, I was using a lot some free software available on internet to perform analysis, but was always very long, tedious, and complicated, and required a lot of passage. While when I started to use the Athena, was really easy and user-friendly, the system, and it was very fast also to have the result for the analysis that we wanted to do for this project. And these allow us to screen more than 130 drugs in very few months, and to have good results and identify the drugs that were more efficient in treating the Kaposiform Lymphangiomatosis disease. So luckily we were in contact with Jason Otterston, that I think is the product specialist of Idea Biomedical, and he was always available via Zoom. So we had, especially at the beginning of the project, we had many meetings to try to understand which of the feature of the fish were more representative for the phenotype, and for us to understand if the drug is what it was working or not, and also to better set like the flow of the experiment, like how we need to prepare the fish, how many fish, and the alignment of this fish in the to get the best result from the microscope, from the Hermes microscope. So okay, the preprint is actually available in bioRxiv, and we are now in the last stage of the revision. We were treating a very aggressive disease in humans that is called Kaposiform Lymphangiomatosis, and currently there are no treatment for these drugs, and the patients they don't respond to the normal treatment. This is an infiltrative disease that leads to enlargement of lymphatic vessels, and the lymphatic system is not working anymore. So what we did, it was first we identify in children a new mutation for one of the genes that is involved in this disease, and we took this gene and we overexpressed the human gene in the zebrafish. So, and we generated a system that allowed us to induce the mutation whenever we want and in the tissue that we want. And in this case, the tissue was the lymphatic tissue. After this, we characterized this model, and we discovered that the fish was presenting the same defects that the humans, making him a good model to proceed and to try to test and identify new drugs. So after this first stage, what we did was to take a library of 130 drugs that are associated with lymphatic disease or with the pathway of this gene, and test them to identify some useful drugs. And in fish that was able to rescue the fish phenotype. And we finished identifying 3 drugs. These 3 drugs were then taken and tested with human cells to see if what we saw in fish was conserved also in the humans. And we finished identifying 2 new drugs that are effective also on human cells directly from the patient. These are primary cells. And they are useful to revert the phenotype. And we hope that with this work, we bring a new possible therapy for this severe disease that is Kaposiform Lymphangiomatosis. It proves that zebrafish present very conserved feature, not only anatomically, but also in terms of pathway with humans. And it means that it proves that it can be a good system to screen drugs. So I think that in the future, we will try maybe more to mimic human disease in fish and to test drugs. This paper will prove and will be like a sort of pipeline, a system, and we can apply this to other disease, and we can use zebrafish to mimic human disease and get some preliminary drug screening that of course will need to be validated in future in mammals like mouse and also in human cells, but it will give us a boost to reduce and to have fast results.